Hello everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode we're going to discuss the evolution of Homo ergaster and the process of fossilization. So let's jump right in. Today's tale takes us to Africa 1 million years ago. Homo sapiens, Neanderthals, Denisovans, and Homo heidelbergensis are yet to evolve. We are firmly among a species of humans likely directly ancestral to us, known broadly as Homo erectus. Homo erectus fossils span from 2.1 million years ago to a mere 100,000 years ago, with the last populations holding out in Java. The earliest erectus fossils come from China. The next oldest fossils date to 2.04 million years ago and come from South Africa. It could mean that either erectus evolved outside of Africa, meaning maybe Homo habilis left Africa first and then migrated to Africa later, or erectus did evolve in Africa, but older African fossils have just yet to be found. The current thinking is that Homo habilis left Africa first, although this does not preclude Homo erectus from having evolved in Africa as well. Either way, some paleoanthropologists prefer to divide erectus into two species: the less derived African erectus, which they deem Homo ergaster, and the Eurasian erectus, which they deem Homo erectus sensu stricto. Dawkins and Wong have opted to use this convention, anglicizing Ergaster into Ergast, but there is some debate on the legitimacy of the name. The reason for this debate is that erectus, including Ergaster, lived for quite a while, some 2 million years, and had substantial variation in anatomy correlating with geography. They ranged from 4 foot 9 inches to 6 foot 1 inch, with a cranial capacity ranging from 546 to 1,251 cubic centimeters. That's greater than the total range of cranial capacity variation in both chimps and modern humans, which is 320 to 480 cubic centimeters for the former, and 1,400 to 1,500 cubic centimeters for the latter. However, the erectus ergaster cranial capacity range doesn't exceed that of some Old World monkey species, like the sexually dimorphic Hamadryas baboon. In general, though, there is a trend in the erectus ergaster complex for a gradual increase in cranial capacity from African and Georgian ergaster to Eurasian erectus. Some experts have advocated for even more species than just erectus and ergaster. For example, remains from Dmanisi, Georgia, have in the past been deemed Homo georgicus, possibly part of an early radiation of ergaster, and 2 hominins from Oceania, Homo Floresiensis and Homo luzonensis, have been suggested to be descendants of island-hopping erectus populations. Whether or not this is the case has been heavily disputed. A 2017 study concluded that Homo floresiensis is more similar morphologically to Homo habilis than to erectus, presenting some intriguing implications, and a friend of the channel, Gutsit Gibbon, has made her own video on the Hobbit of Flores, link in the down part. Or going the opposite direction, where does Homo habilis end and Ergaster begin? Similar arguments have been made around Homo habilis and whether some fossils assigned to it should be classed under Homo rudolfensis instead. The biggest differentiation between Homo habilis and Homo rudolfensis appears to be the brain case, with Homo rudolfensis being more endowed and relative orthognathy. There is such a smooth gradation from one species to another, as expected under evolution, that trying to divide populations into separate species is understandably difficult. What about ergaster morphology? Aside from having a smaller cranial capacity, erectus was more robust than Homo sapiens, with larger molars, a larger brow ridge, and a more prognathic face. Facial prognathism gradually becomes more apparent as we approach our common ancestor with chimps, though we have about 5 million more years before we reach that point. The specific name for erectus refers to their gait. Erectus walked as erect as we do. Homo ergaster, on the other hand, means working man. There is the interesting matter of how hairy Ergaster was. We have a protein called melanocortin 1 receptor, or MC1R, that is involved in regulating the pigmentation of skin and hair, the gene for it being expressed in melanocytes. The gene for MC1R has 10 amino acid differences between the last common ancestor of humans and chimps, as well as a number of differences between non-Africans, but 0 amino acid differences in modern African populations. A 2004 paper argues that this indicates strong selection on the gene after our common ancestor with chimps, but before the split between Africans and non-Africans. Given the observed genetic diversity of the MC1R locus, the researchers calculate that the strong selection occurred about 1.2 million years ago, while Ergaster existed. The researchers hypothesize that selection may have resulted from a combination of our ancestors spending less time in forests and more time on the savanna, as well as from reducing our hair coverage for more efficient thermoregulation. These factors placed pressure on our ancestors to adapt resistance to UV radiation, which they did by darkening the skin. This pressure was later relieved when Homo sapiens left Africa. However, the pressure would have been reapplied when Homo sapiens returned to regions, especially equatorial ones, with intense UV radiation. There is another pressure responsible for skin color, biosynthesis of vitamin D3. This vitamin is, quote, important for the regulation of calcium and phosphorus metabolism, skeletal development and mineralization, the regulation of normal cell growth, and the inhibition of cancer cell growth, close quote. A chemical called 7-dehydrocholesterol, which can be found in the keratinocytes of the epidermis and the fibroblasts of the dermis, is stimulated by UV photons to become previtamin D3. From there, previtamin D3 undergoes spontaneous isomerization, meaning the arrangement of atoms in the molecule changes without adding or subtracting atoms to become active vitamin D3. So, how did this process contribute to the evolution of pigmentation? One of the jobs of melanin, which if you remember is regulated in part by MC1R, is to scatter light, protecting the skin. As humans moved out of Africa and into regions with less UV exposure, having deeply pigmented skin, which was beneficial in Africa, would have become detrimental. Therefore, the production of vitamin D3, which is regulated in part by UV hitting the skin, would have placed a selective pressure on non-Africans to lighten up. These competing pressures have generated the modern suite of skin colors. But morphology wasn't the only thing that evolved. Tool usage evolved too. According to a 2014 paper, quote, thus, the increase in average body and brain size from Australopithecus to early Homo to Homo erectus is consistent with a greater control over, or amelioration of, mortality risk and increased nutritional sufficiency. Increasing cultural mediation and enhanced niche construction through technology and social factors such as food sharing or allocare would be essential to buffer against fluctuating climatic conditions, reduce predation pressure and extrinsic mortality risk, and ensure greater food availability, close quote. One technology that appears to have been used by Ergaster was fire. Though it is uncertain when hominins first started using fires, which was likely more than 1.5 million years ago, fires at the time would have been initiated solely by lightning. From 1.5 million to 300,000 years ago, fire usage is sparse. Perhaps hominins carried fire from its natural ignition sites, a sort of transitional stage in fire use. But from 300,000 to 70,000 years ago, the burning season appears to have significantly altered, with humans starting their own fires, being apparent in the increasing amount of burnt flints. The usage of fire only grew from there as humans spread across the globe and later incorporated fire in agriculture. It is likely that tool use on processing animals predates fire usage. Fire's immediate benefits may have been more related to protection from other animals on the savanna rather than cooking meals, which may have been more of an added bonus. Here, perhaps biological and cultural evolution entered a positive feedback loop. Tool usage and long social histories already existed in primates, and humans merely built upon these. Simple tools like flaked rocks could have helped hominins kill and process animals more easily, allowing hominins to incorporate larger amounts of meat into their diet. larger amounts of meat, and especially cooked meat, meant that humans got more calories through their diet than their proportionally more herbivorous relatives. One reason is that cooking food means less time is spent digesting the food, allowing more metabolic energy to be allotted to other processes. As a result, humans have biologically adapted to increased use of fire. According to a 2010 paper, quote, "The evidence that the average woman eating a diet of 100% raw high-quality foods is amenorrheic suggests an important conclusion. Human populations are not adapted to survive on a diet of raw wild food, even when it is extensively processed using non-thermal methods." This idea is consistent with the fact that no human populations have ever been found living on raw wild food, close quote. The paper further writes, quote, "Even though honey, marrow, liver, and some exceptional other kinds of meat or fruit or social insect might in theory sustain a population when eaten raw for a few weeks or months, we know of no raw diet that could provide predictable year-round adequacy," close quote. Other morphological adaptations include a shortening of the colon and cecum, since long-chain carbohydrates didn't need to be fermented, a reduction in molar size and jaw muscle myosin, an increase in salivary amylase production, and, possibly, an increase in brain size. As brains are metabolically expensive organs, an increase in caloric consumption could have allowed them to get larger. The ultimate pressures for larger brains in primates, and consequently hominins to a much more significant degree, is currently fiercely debated over. It is important to remember that ultimately the pressure was likely multifaceted and involved elements of sociality, or relationship management, ontogeny, both in vitro and the hominin juvenile stage, and diet. It is also worth asking how our gut microbiota changed as a result of incorporating higher concentrations of meat into our diet. While our gut microbiota is, unsurprisingly, most similar to the other great apes, ours contains a higher proportion of taxa associated with diets containing animal products. The shortening of our colon and cecum also meant there was less room for a diversity of microbes, and human coprolites from the past 8,000 years have indicated a shift in microbiota towards that of modern rural communities. A 2015 paper writes, quote, "Calcified dental plaque documents compositional shifts and declining diversity in the oral microbiota during the transition from hunter-gatherers to the Neolithic, and again around the Industrial Revolution," close quote. More recent Homo remains have a more carnivore signal indicating more meat consumption. For reference, chimpanzees track with herbivores despite a portion of their diet consistently involving meat. Researchers can determine the diets of animals by looking at the proportions of certain stable isotopes like carbon and nitrogen in their feces, hair, and enamel. Now let's open one more can of worms: the evolution of language. This topic has been so controversial for such a long time that the Linguistic Society of Paris in 1866 and the Philological Society of London in 1872 prohibited their members from making speeches on the subject. In the past 50 years, linguistics has been married to evolutionary biology, resulting in a field called biolinguistics. One major concept that has come from biolinguistics is universal grammar theory, which is typically attributed to linguist Noam Chomsky. To quote resident Ceratosaur Dapper Dinosaur, quote, "Humans are hardwired to divide the world into discrete entities, and then talk about things that those entities do, or that are done to them, and we split these actions into their own discrete categories, and we tend to have cross-linguistically similar, but not identical, categories, which is why most nouns and verbs in one language correspond closely to a word in another language," close quote. In other words, human languages generally have certain structures, which is the result of constraints of our brain. Possibly, as has been suggested by anthropologist Ian Tattersall, quote, "The neural capacity for language was acquired with the emergence of Homo sapiens 200,000 years ago, but that this new potential was not exploited until about 100,000 years ago through processes of exaptation and emergence," close quote. Other researchers have posited that things like the ability to understand the thoughts of others have roots in non-human animals, meaning that some aspects of language could have gradually assembled in our ancestors before merging together. This is supported by a myriad of theory of mind studies on the great apes and Old World monkeys. Further, the role of gestures is also considered to have been crucial in the evolution of language. It should be noted that human toddlers and chimpanzees share upwards of 90% of their intuitive gestural repertoire. Humorously, one of the most famous signing animals was a chimp named Nim Chimpsky. Though one question we have to ask is, could any of our hominin relatives speak in the sense that we, as modern humans, can? In the instances where we have good paleontological records, the evidence tends towards probably not. Neanderthals have been debated back and forth. On the one hand, they had a basic cranium that argues against speaking, but on the other, preserved hard portions of the hyoid apparatus seem to argue in favor. Whether Ergaster could speak is completely unknown. Regardless of the exact path of language evolution, a gene called FOXP2 seems to be involved. FOXP2 is not the gene for language, despite what some journalists have claimed, nor is VMAT2 the God gene, but that's another story. Rather, FOXP2 is a transcription factor that regulates the expression of genes involved in the brain, lungs, heart, and digestive system. Homologs of FOXP2 exist in jawed vertebrates, and as one might expect, this gene also plays a role in both bird songs and bat cries. Both birds and bats go through babbling stages of vocal learning, just like humans. However, the extent to which bird, bat, and even other primate sounds are homologous to those of humans is debated. Our FOXP2 differs from that of other great apes by 2 amino acids. However, whether those 2 amino acid changes are what confers the complex language ability in humans is debated. Interestingly, Neanderthals and Denisovans had the same FOXP2 as modern humans, but both species lacked a number of other alleles involved in language, such as the human versions of CNTNAP2 and ASPM. Experiments with FOXP2 have had intriguing results. For instance, mice given the human FOXP2 gene made vocalizations different from mice without. The mice with the human gene could reach higher peak frequencies than those without. Indeed, far from being the gene for language, FOXP2 and other transcription factors merely provided the capacity for language to develop. Further, those 2 amino acid changes may not have been directly selected in hominins for the language capacity. According to a 2014 paper, quote, "Since FOXP2 also targets the gut lining, the evolutionary step may have had little to do directly with language, but instead with digestion modifications driven by forest to savanna habit, and so dietary change, as in the well-established case of lactose tolerance and dairy culture." Close quote. Speech in the human sense is certainly unique within the animal kingdom, but this should be appreciated as only one type of complex communication. To consider human speech as the pinnacle of communication is to ignore the intricate vocalizations, gestures, and chemical signals that can be found in other species. Human language can perhaps be best understood as a natural evolutionary step for a species so invested in sociality and communication, allowed only thanks to a fortuitous increase in areas of the brain conducive to sending, receiving, and processing information. Suffice it to say, there are still a lot of outstanding questions with regard to the evolution of language. More generally, there are a lot of outstanding questions with regard to both cultural and biological evolution within Ergaster. A main part of the reason is that culture fossilizes very poorly. Much ink has been spilled over whether Neanderthals had their own version of religion, a topic we are not going to touch as this video is already long enough. Painted shells, bits of cloth, and charred flints provide the fodder for much academic debate, but the archaeological record in many instances is so spotty that only speculations are produced. That's not a bug, but a feature of the fossil record itself. You see, fossils form only under particular sets of circumstances. Minerals are made of repeating molecular motifs. Calcite, for instance, is formed of repeats of calcium carbonate, quartz is silicon dioxide, diamond is pure carbon, corundum is aluminum oxide, and so on. We even have minerals in our bodies. Calcium hydroxyapatite is the primary mineral involved in our bones. Rocks, then, are solid composites of minerals and/or mineraloids, the latter of which includes amber, obsidian, and diatomite. There are three types of rocks: igneous, metamorphic, and sedimentary. Igneous rocks are cooled magma and lava, including granite, gabbro, and diorite. Metamorphic rocks are those that have undergone intense heat and pressure, resulting in physical and chemical changes, including quartzite, gneiss, and marble. Finally, sedimentary rocks are those that have formed by the deposition and cementation of minerals and/or organic particles, including sandstone, limestone, and breccias. Sedimentary rocks are the rocks most interesting to paleontologists, as these are the ones that contain fossils. Fossils then come in a variety of types, from body fossils to negative impressions to footprints, coprolites, and amber fossils, the latter two of which we've discussed in our paleontology playlist. Fossils are the remains of organisms that died at least 10,000 years ago, and they inform us about life in the distant past. Hard parts like bones and shells fossilize more easily than soft tissues like organs and feathers, because they are less susceptible to being destroyed by heat and pressure, meaning hard parts make up a much larger portion of the fossil record than soft tissues. That's not to say we don't have fossilized soft tissues, we do. These most prominently come from exceptional fossil deposits called Lagerstätten. The limestone Solnhofen Formation in Germany, from which Archaeopteryx hails, is one such example. Amber also often does a really good job of preserving soft tissues. Trace fossils, also known as ichnofossils, are sometimes preserved too. These include burrows, footprints, and coprolites, and are records of the lives that organisms lived. An interesting recent trace fossil from South Africa contained elephant trunk drag impressions. A fossil doesn't just tell you that something died, it can often tell you how something lived. This video has contained numerous examples of that. Organismal remains are left in the sediment, and these can become converted to fossils through various ways, often involving rapid burial or, at the very least, sequestration from destructive forces. For example, bone hydroxyapatite is often replaced with surrounding strata minerals, leaving behind a rock in the same shape through a process called permineralization. That's a topic we'll have to explore in more depth at another time. For now, we'll leave you with this. Organisms and their remains have the potential to end up as fossils in sedimentary rocks, but organisms rarely fossilize due to various geologic processes. As a result, much to-do is made when fossils are found, as you have no doubt noticed. So, with both the evolution of Ergaster and the very basic principles of fossilization covered, we are going to sign off. So, thanks for watching, and we'll see you next time.